welcome to another one of our interviews. Today we're doing a very special interview with some early career researchers who have published the first paper with us in Acta Cryst F. I'm John Aguirre from the University of York, and joining me today is Graham Chakafana from Hampton University in the US. Hey Graham, how are you doing?
Hey, how are you, John?
So today we're interviewing Sydney. How are you doing?
I'm good. How are you?
And Alex.
Hey, how you doing? Pleasure to be here.
Great to have you. Right, so we're going to get straight to the questions. And the first question is Graham's.
So my first question to either Sydney or Alex is, what drew you to this particular research project on NMTs?
So for me personally, I've always been really interested in drug design, drug discovery research. So this drew me to the topic, and specifically for the project, the prevalence really caught my attention. There's definitely a need for new therapeutics, especially for Plasmodium vivax malaria, and Plasmodium vivax being the second most deadly of the Plasmodium species. So the treatment options currently are really limited, so this research is very necessary.
Thank you, Sydney. Alex?
Yeah, pretty much just going off what Cindy said. I think with science, you always need to have a good story, and that's what caught my attention with the Plasmodium vivax malaria. Hearing about how 2.5 billion people are at risk because of antimicrobial resistance, and like Cindy said, there's only one drug in the market that's Primakin. Once Plasmodium vivax is able to evolve and adapt to this drug, our last line of defense is gone. So, finding more drugs is crucial for this battle.
Okay, thank you, Alex.
Yeah, next question is mine. These will depend on how you divide the work amongst yourselves, but what analysis do you perform during this project?
Pretty much me and Cindy and the rest of our lab mates, we all kind of fell in line naturally. Some took the approach or the challenge of using the primal as well as chimera to understand the structure-based analysis of the drug and the protein interactions and all the crucial key amino acids binding to it. There's other people that were involved with the multiple sequence alignments, trying to observe conserved domains and differences, overall trying to help contribute to the process of creating a more efficient drug.
I mainly did my work in primal, so looking at the three-dimensional structure, and also a cluster omega, being able to do that multiple sequence alignment to compare the different amino acid sequences of the P-vivax to other isoforms of the NMTs like PvNMT, which is Plasmodium falciparum, which is the most deadly of the Plasmodium species. Also compared with the human isoforms of the NMTs as well.
Ah, fantastic. Thank you.
Okay, so I have the next question then. So what aspects of the study did you find most exciting for you?
I was a bit intimidated by bioinformatics and computational tools. So I think that now becoming proficient and comfortable with these types of tools was really exciting, and being able to make the figures for the paper was really rewarding as well.
And Alex, what did you find most exciting about this work?
Pretty much from what I've learned in my first 2 years of undergrad is you can't avoid the computational sciences anymore. So this provided a really good opportunity to learn directly with the computational tools that are used to study protein interactions or protein drug interactions. So this is a very good learning opportunity that could help me advance within my career.
What opportunities and challenges exist in designing inhibitors for Plasmodium vivax NMT?
Yep. So one of the opportunities that could definitely be explored right now is we've performed structure analysis on this Plasmodium vivax NMT inhibitor. However, it hasn't been used directly to treat Plasmodium vivax. So there's definitely opportunities to explore the in vitro and in vivo studies to observe, if there's any off-target effects, is it entering the cell correctly, and so much more.
And Sydney?
So as far as challenges, I think Alex mentioned this earlier, but antimicrobial resistance is a really big problem. And so this would be an opportunity to create new drugs um, to combat the vivax malaria. Yeah.
So my next question would be, what other challenges did you personally encounter when you were conducting this project?
Personally, definitely getting familiar with the computational tools, and I also use a MacBook, so a lot of the software isn't very compatible on Mac. So bypassing, just figuring out how to work those different softwares was definitely a challenge for me.
Thank you, Sydney. And Alex?
Yep, some of the challenges is was I just never, worked directly with proteins. So being able to be a sponge and consistently obtain and retain information, but not only just being told what to do, but also help contributing by being an independent scientist. So this definitely helped my skills in critically thinking on my own and giving suggestions to Sydney and Dr. Shakafana on the work.
Thank you.
How do crystallography and structural biology contribute to the drug discovery and development process?
Structural biology, structure and function have a direct correlation. So being able to look at the three-dimensional structure of proteins, you can see the different binding pockets. And especially for this project, seeing how the inhibitors bind to the NMT, that gives a lot of information about the function and if this could be a viable inhibitor for the NMTs.
Cindy explained it pretty perfectly. It gives you a very high resolution, structural confirmation of how the drug is binding towards the active site of the Plasmodium vivax NMT enzyme. So gives you a lot of information that you can use to make modifications towards the drugs. But also, it can be applied in other sciences such as protein-protein interactions for, cancer and other sort of things.
Perfect, thank you.
So now let's talk about the process of writing the paper. So since this was your first research article for both of you, how was the process? What were some of the challenging things, or what advice would you also give people that are trying to write their first manuscripts, like undergraduate researchers that are also trying to write their first manuscripts?
Yeah, one of the biggest challenges for me was just learning how to read a paper. I think that is one of the hardest skills as an undergrad is you have a lot of papers out there and a lot of jargon that definitely confuses you. So learning how to build the skills and knowledge to efficiently read a paper um, and use that when you're writing. And another one that I had was time management and learning to communicate with the other members of the lab was a really important skill that I learned from this opportunity.
I totally agree with Alex. This was really the first time that I had read scientific literature, and I found that, like Alex said, the jargon was hard to understand at times. But really, reading papers, I would say, is a good advice, learning how to read scientific papers, because it's a really essential part of understanding.
Okay, thank you.
What new skills did you gain through these projects, and how might you apply them in your future career?
Yeah, so I definitely gained a basis and a foundation um, to understanding computational work and bioinformatics. And I've taken this foundation and I've applied it to other projects that I've done. I've done projects with crystallography, and I've already understood the research and why crystallography is so important. And I've also done projects where I attempted to do molecular docking, and I definitely wouldn't have attempted that if I didn't have the foundation that I built from these works. And I plan to do structural biology research in the future as it applies to, drug design and drug discovery as well.
Right, so the choice of project had an influence on your aspirations for your career path. That's
Yeah.
excellent to hear. Right, Alex?
Yeah, for me, gaining the foundational skills in bioinformatics as well as understanding protein targeting and protein, in general actually gave me the opportunity to work at a biotech company known as Cargo, where instead of applying it such as antimalarials or other antimicrobial, directions, I actually applied it in the food science where I was using these skills that I learned, but instead working on it for on an enzyme for sugar conversion.
Very interesting. Thank you.
So, do you have any advice that you would want to give to people that are hoping to publish manuscripts or do this type of work? What piece of advice would you give those people?
I would say at your home institution, look for labs that are doing research that you're interested in. Talk to students in that lab and see what it's like. Talk to the PI of that lab as well. And also, if they have a lab website, I would definitely check it out and see if they publish as well.
My piece of advice for undergrad students is, just to not to not be afraid. When I was looking for research, I really didn't know what I wanted to do, but I went to Dr. Shakafana's lab and I just asked, hey, what was your research? Is it possible if I could join? And by having that just one little conversation, I was able to get these foundational skills that now I can apply to my first year of education at my PhD program here at the University of Virginia. So all these tools and knowledge that I gain are actually helping me and giving me an advantage here at the University of Virginia. So always communicate and look for new opportunities and just to learn.
Okay, thank you, Alex.
We've come to the end of the interview. Thank you very much, Sydney, Alex, for doing this interview with us. And thank you very much, Graham, for being such a fantastic interviewer.